Okay, so as we introduce topic F2, which has to do with virology in this case, we are going to have a little bit of a sub portion of it at the end when we deal with prions, virinos, and viroids and things of that sort too. But we'll save that for the second part of this recording. Now, going on with it, as always, there's the material that I want you guys to study, so your SLOs. And one of the key features to understand viruses, especially with the topics that are going on right now, is how do we categorize viruses? How do we classify them? But more than anything else, before we can classify them, we kind of need to know what they are. And even then, up until very, very recently, people had some disagreements associated with it. So we're going to talk about what we call living criteria. How do we declare a virus, whether it's alive or dead, and so on and so forth? And then we'll definitely get into the classification, which is something you definitely need to know, right? And then there are various methods of infection. How do they get in? How do they attack? How do they take over? And then part of that meaning taking over the system, hijacking it, making more copies of themselves. So it's this idea of replication. And then towards the end, that topic that we addressed with the HPV subject is this idea of how can they trigger things like cancer? How can they cause cancer? So we'll go on to that towards the end of this portion. So let's get with some definitions. Part one of this story, right? So far, everything we've covered all the way from topics A up until now has been dealing with things that we've called alive. This idea that these organisms are made up of cells, they have some sort of nucleic acid, some sort of DNA of some sort in the tiny little container. And all of these organisms, as we've covered in their topics, have these abilities, whether it is to grow in their number or in size. They have this ability to respond to their environment, to consume things and process them, and to use their nutrients to either grow and divide or also produce wastes, all of that kind of connected. And then they had a little bit of a pattern of organization too, kind of how you build from 1 cell to 2 cells to 10 cells, whatever it is, into tissues and so on and so forth. All of that applies until you hit viruses. And so one of the things that has happened is because viruses are not made up of cells, immediately the definition became viruses are not alive. Oh, I'm here to change your mind. So for those of us who kind of thrive and have some fun in the fields of virology, we don't hold that same definition. So for our class, we're going to adapt with that. We're going to go with the one we understand best. When people talk about things being alive or not alive, remember, humans, us, we happen to be more a little bit on the egocentric side. We like to compare things to us. And so when we talk about a dog, when we talk about a cat, we go, well, that thing has 4 legs, so it has 2 more legs than humans. Well, that's not very fair. And even though there's tons of humans on the planet, that animal had 4 legs to begin with, right? And even though they're kind of armish and kind of leggish, why are we comparing it to us? Why are we the center of what we describe on this planet? We haven't even been around on this planet that long, right? So we call this a top-down view, meaning that we consider ourselves to be on the top of the definitions, and then everything else on the bottom has to fit us. And so when we do that top-down view, one of the things that happens is the virus doesn't fit in, right? Because it kind of challenges the definition of any biological living organism at this point in time, because it doesn't, for example, breathe. It doesn't partake in respiration. It doesn't, transform any other substance, whether it's oxygen or not, into some sort of energy pathway. It doesn't really respond to its environment like most other organisms that you poke them and they do something else. Doesn't get bigger, which is kind of interesting. It does grow in number, but not in size. But it does perform certain other tasks. So what virologists and microbiologists have done with this topic is kind of changed it around, saying, well, rather than trying to compare everything to humans from this kind of top-down view, we decide, well, why don't we start with the smallest thing? Why don't we go from a kind of bottom-up perspective? In this idea, we're saying, well, what is the most simple, basic, tiniest thing that we have that we can consider to be alive? And if we do that, viruses happen to fit that specific definition. In other words, what we're saying here, it is the most simplest organism that we have that is capable of reproducing, of making more copies of itself, right? And it can indeed, more than anything else, adapt to its environment. As you all know, as we've been seeing lately, specifically with the viruses floating around, that they can change, they can improve, they can hit a little bit harder. And so those are typically traits of something that is alive. So with that main idea, right, rather than kind of considering everything living having cells, well, this is an acellular organism, no cell included, right? But it does contain a little packaging system, a little wall, a little container, just like all cells do, made out of phospholipids. This one's made out of protein. And it definitely does have nucleic acids inside of it, just like every other living thing that we know, right? And now the only requirement that viruses have is that they need to be inside of a host. In other words, they have this obligate replication requirement so that they can make more of themselves. So they need to be inside a cell to do so. So rather than kind of looking at living things kind of as a snapshot in time, just saying, well, right here, right now, we define this as this one thing. Instead, what we can do is say, well, if we look at viruses over hundreds, thousands, millions of years, we can actually see that they've been around even before us, right? And they've been adapting, changing, becoming stronger, faster, and so on and so forth, and still growing and making more of themselves, like any other organism would do that. So we start kind of providing a new definition of what life can be. And so by that definition, we can pretty much state that viruses are also alive. They are living organisms under certain conditions. In other words, they require a host to be able to do all of their properties. That's kind of where our definition goes. So we look at the definition of a virus as a lineage, not as just right here, right this second, but rather over time. And then if we include that definition over time, basically what we're saying, it's evolution, is that these organisms can grow, can do their thing, they can adapt, they can respond, that they can take over, they can manipulate, so they can respond to their environment, they can change things, they can kill things, they can use their systems just like any other organism on this planet does, or most of them can do, right? So when we were talking about classification in the earlier topics, that's when we talked about things like some of our alpha proteobacteria, like the rickettsias, and we talked about certain febrile diseases like typhus and so on and so forth. Those were also tiny little cells that were infecting others. These were bacteria instead. And they were able to infect, to get in, to hijack other systems, very much like a virus, even almost in size. So the only thing that ends up missing here is that the difference between things like rickettsias and coxielas and things like that versus viruses is that phospholipid component, the little cell component. So if you take that kind of out of the equation, they both behave pretty much the same way. They kind of get in, they hijack, they kill, and then they go and make more of themselves. So they do pretty much the same type of pattern. So if we go with that again, right, we can kind of have a summary of all the properties that a virus does have. Well, sure, a virus cannot perform, in this case, metabolism on its own. But the second it's inside of cell, no limits. It cannot reproduce independently, right? While it's floating around outside of a host, not doing much. Second it gets inside, hijacks the system, and can do all of that. Takes over. Now, there are no organelles, right? So there's no flagella, there's no ribosomes, there's nothing like that. But again, the second it gets inside the cell, it can do it all. It takes over all of those and uses it at its will. So as we start seeing that picture of what we define as a cell versus an acellular organism in something being alive or not alive, the viruses fit really, really well within that definition. The only thing is we have to kind of change our perspective. Stop thinking that humans are at the top of the food chain and that we run the show, and instead start understanding that the most basic, most primitive, smallest things out there are still capable of doing a lot of the things we do, just with some requirements, some minimum properties so they can do their thing. Now, the part that will change for today, and we will discuss this as we go further, is that they don't quite follow the central dogma as we do. So far, everything we've done, as we perform in the previous exams too with the central dogma, is that whole DNA, RNA, protein, and that's how pretty much everything we've covered so far works. Today, we're going to actually change that. We're going to look at it a little bit different. So let's start with that. What makes our virus a virus, right? So let's start with some properties. What do they have? Well, in this case, they're all submicroscopic, meaning nanoscopic, if you will, right? They're about 1,000 times smaller than most bacteria, even though there are some giant viruses out there. So there are some exceptions to that, right? They have no lipids outside of them. They're acellular, so there's no lipid bilayer like we learned during topics C, for example. They don't have any, lysosomes or ERs or anything of that sort included in them. They're just kind of efficiently packaged. And what's unique about them is that they're infectious. As we will get into topics I and J eventually when we talk about transmissibility and infection and something being contagious, we'll expand upon those definitions, right? But our particular one, by a virus, that means it can be transmitted, and it must infect, meaning it has to land on something and get in and stay there, okay? And now there will be variations on how much is needed for it to stay there. So things like viral load and these definitions will kind of come in a little bit later, all right? So now we also start with being able to tell the difference between what's outside versus inside. And so most of us use the common terminology, we just call it a virus. But in reality, a virus is the term we use when it's inside the host. When it's outside floating around doing its own thing, we call it a virion. And this is very unique to microbiology and virology. Pretty much everybody outside of this field just calls it a virus and that's it. For us, as we become a little bit more experts in our field, if you will, we know that while it's outside the host, it's called a virion. When it gets inside, when it hijacks, it's called a virus. So be aware of that difference in there, okay? So the extracellular version of this, the virion, typically has its own little container, this little protein coat that builds up its little housing that has a fancy name called a capsid. And this capsid, when you put it together with its own genome, with its own nucleic acid, can be often called a nucleocapsid. Sorry for my enunciation there, right? And it is this virion stage, this external stage of both protein coat or protein encasing and its nucleic acid inside that is the infectious portion. That's the one that is transmissible. That's the one that gets passed on, okay? That's the one that's actually needed for you to be able to infect another host, to find it and be able to get in. As you will find out, the other stage, which is the intracellular version, the one we called an official virus, is typically just a nucleic acid, meaning just the DNA or RNA, as we'll talk about in a moment. There's no capsid present, okay? And that's the part that hijacks, takes over, and does its thing. That's the one that's manipulating the system. So the key difference here, though, is that external version, that capsid version, that container version, in this case, is needed to be able to infect. If the virus was just floating around with just its nucleic acid, it wouldn't get in. It doesn't infect. It can't be taken into the cell and do its thing. So it actually requires that external version. It requires that casing, that outside layer of protein, that capsid that we're talking about, to be able to get in. If it's just kind of floating around without its capsid, not infectious at all, as far as we know, mind you. There are some weird exceptions that appear in plants sometimes too. So let's kind of do a quick comparison. And this one I've taken from your textbook directly, so you can always look at the summary. This one does a pretty good job of using a little bit of a table and kind of summarizing all of this, right? It takes the difference between what cells or cellular organisms are versus acellular organisms like our virus, like what they can do and cannot do based on their conditions, right? So as we continue with our definitions and our classification, specifically with viruses, well, now let's enter the categories required to kind of describe where viruses fall. where do viruses fall in this case? And so let me give you a little bit of background in this case. What we have here is that viruses, since their discovery and the scientists, all the awesome scientists that study viruses kind of gotten together, a committee was established called the International Committee on Taxonomy of Viruses back in 1966. It's called the ICTV. This case got together as a way to create the same types of categories that we use in standard taxonomy and classification. We've tried to design what we call a universal establishment of how to sort these out, just like we do with fungi, just like we do with protists, just like we do with all our previously studied organisms. Problem is, we didn't have enough information. One, relatively new field in comparison to the other ones, but also not a lot of information, especially when we're trying to study them. As most of you probably will agree, nobody wants to be a volunteer for Ebola, so we can study Ebola a little bit better, right? And so it makes it a little bit more challenging, especially when we know that these guys are really effective and some of them can be really deadly. So the amount of information that we have, as you know, fell into the three domains that we've talked about in the previous classes, our eukarya, our bacteria, and archaea. But viruses did not really belong, not only because of the fact that they were considered not to be alive, and so therefore did not fit within that kind of cool tree of life that we talked about before, but more than anything else is that we didn't understand them. And so we actually had the system of classification until recently that made it only as high as order. We didn't have any phyla, we didn't have any classes, we didn't have any kingdoms, or in more than anything else, we didn't have a domain. Again, no life declared, so they didn't quite fit with everything else. So since the 60s, at this point in time, we could only classify viruses limitedly. Even though we knew enough of them, we couldn't quite find out where they fit beyond that level of order. And so each one of those has a little bit of a rule. Every taxon that you see in there, from order to family to genus and species, and some of them include a subfamily, all end up with the same name. So if you end up hearing something that ends in the term Virales, that means it's an order. If it ends up with Viridae, it's a family. If it ends up with Virinae, it's a subfamily, and so on and so forth. And only once you get to the bottom of genus and species, and sometimes even subspecies, you get the standard names that most of you are familiar with, like, measles virus and hantavirus and Ebola and so on and so forth. That's where they get that low. Okay, so be aware of that, because I'm actually going to give you some information a little bit later that is probably new and improved, something really cool. So how do we do that whole classification bit? What properties do we use? Well, they don't have a cell, they don't have a wall, so we can't use any of those properties. But what we can use in this case is their genome, their nucleic acid. That's the number one way, and I'll get into that one next. We can also classify them based on what they infect. Who's the host? What is the host? If you will, do they infect plants? Do they infect humans? Do they infect both? Do they infect fungi? And so on and so forth. So we'll look at those as well. And then another way we use to classify them is their shape, right? What shape do they take in terms of their cool little capsid, that cool little protein coat that contains them, the container? Based on the shape they take, we actually can classify them as well. So those are the properties that we use. And then when all of those kind of overlap, because they do, in case you're not familiar with, for example, there's an estimate of around 300,000 to probably 1 million mammalian viruses. So let that sink in for half a second there too. So a lot of them are going to overlap. So how do we sort them out, right? So every now and then when they do overlap, well, we also start looking at the diseases that they cause, what type of symptoms did they produce, what type of immune responses do they elicit from us, what type of proteins do they carry. All of those are kind of subcategories that we can use to classify and taxonomize these guys. So those are the top three that you should be familiar with, but all these other ones can be used kind of off to the side when we want to learn some extra information. So let's start with the first category, okay? Our very first one, the genome itself, the nucleic acid, the blueprint, the instructions that we were talking about before, right? Well, this is always an important one because in this case, size matters again. So what do we have here is the fact that we can take the several cajillion bases that humans have. Yeah, I'm being funny in this case. So the 3 billion bases that every single cell in humans have, and compare it to, for example, a bacterium. And the one you're seeing over there is E. coli. And so if you see E. coli on the lower or on the left-hand corner over here, you see all that little string that you're seeing kind of floating around. That is just a piece, a partial chunk of the nucleic acid of the DNA that E. coli has. Okay, so if you do like a little bit of pictures, you notice that it's about a quarter of it. Now, if you pay attention to the top right-hand corner, so I'm going to use my little doodler over here as usual. Let me get this guy going. Actually, allow me just to use the laser pointer. If you pay attention over here on the top, you'll notice where it says viral genome. And you can see how tiny that thing is. And it's that guy is the one that is capable of hijacking and taking over, right? Now, I can tell that, for example, most of us can kind of package up about 6 feet worth of DNA in each one of our cells as humans. But E. coli's DNA is about 2 millimeters in length, roughly speaking. Okay, remember that E. coli is about 2 microns in size, so much smaller, obviously. But it's just tiny, tiny, tiny. Chlamydia, for example, Chlamydia trachomatis, one of the ones we get to talk about later on, only has about 800 genes. However, when you look at something like MS2, one of the bacteriophages, one of the viruses that infect bacteria, that's 3. That's a huge difference in the amount of DNA. It only needs 3 genes, 3 little pieces of code, 3 little instruments of proteins to take over and destroy its host, to literally get in, hijack, make more of itself, and take over. So size plays a huge role here too. Now, the other piece is, well, what type of nucleic acids does it carry? And this is kind of an interesting one. Remember, I told you that viruses don't quite follow the central dogma the way we normally studied it. Viruses can carry DNA, as we normally do, but they can also carry their instructions in just RNA form, flat out RNA. So one of their advantages is that it can start a little bit earlier. So viruses can come in either DNA versions or RNA versions. Never quite both, as far as we know so far, mind you. Okay, and again, reminding you that the amount is really, really tiny. But another twist into this story is that we know that, for example, our DNA is double-stranded. You know, we have our 2 strands of DNA. One is our coding strand and one is our complementary, and so on and so forth. These guys don't have to do that. Sometimes they have 2 strands. Sometimes they come with 1. So they can be single-stranded DNA or they can be double-stranded DNA. And the exact same thing applies to RNA as well. They can be single-stranded RNA viruses or they can be double-stranded RNA viruses. Okay, so they can appear in both versions. And even more so, not only in terms of their strandedness and whether they're DNA or RNA, but they can come in multiple copies. In other words, they can come in chunks of their blueprint, kind of like us in our chromosomes, in which we have several different pieces of them. Well, they can do the same thing. They can have one that encodes all their proteins, or they can have multiple pieces that encode all their proteins. And then also, in terms of their arrangement, very similar to bacteria and things like that too, they can be all linear or circular. Just like bacteria do have their circular DNA, and mammals and humans have their linear DNA, they can do both. I mean, either or, I should say, not both at the same time, right? And then the one piece that we end up talking usually during the central dogma lecture is the fact that their DNA or their RNA can come in terms of their ability to decode it, meaning they come as what we call positive sense or negative sense versions, in which they require an extra step for them to be able to make protein. And we'll kind of give some examples a little bit later. All of these are variations. So when you look at the bigger picture, you'll notice that there's this huge amount of classification that exists in terms of how do we sort these guys out, right? So not only they can become DNA or RNA, they can become double single-stranded, they can become linear or circular, and they can come in multiples of single pieces, but also in terms of the their decodability, which we'll talk about next. So there's a vast amount of information that is available on just trying to sort out most of these viruses. So as part of the classification, one of the things that we do want to expand is that decodability. So what does that mean to us? And so I'm going to try to kind of exemplify it in a very simple version, and we can expand upon it a little bit later, is that most of the things we've learned so far, in this case, is that everything follows this so-called positive sense, in which when we're starting to give examples, let me get a little bit something for color over here. There we go. We all started with our double-stranded DNA, right? We have our 2 double strands that you can replicate and make more, right? Make 2, copies of those guys. But ultimately, you would have to go into transcription, and through some awesome polymerases, you'd make a single strand of that guy, right? And then eventually, under the right conditions and everything else, you can enter translation, right? And using some cool ribosomes, you would make some cool little protein, right? Something of that sort. And that was the path. That's the central dogma that we all learned, right? DNA, RNA, protein. The instructions, the decode, and then the functional piece. So far, that's what we did with our second exam. And that's what we call a positive sense, in which the instructions are ready to read, ready to use, ready to go. But what happens with this so-called negative sense that we were just mentioning a little bit earlier? And so this is going to be a more important property, especially in viruses that can do some very sneaky things. A negative sense typically requires an extra step. So I'm going to kind of erase everything that is in here. And I'm going to show you that comparing to, for example, the original DNA, remember that you had 2 strands of DNA. I'm going to separate them out in this case. And then we only read one of them, the one that ultimately would give us the RNA that we needed. Let's do this again. That guy would give us the RNA that we needed, which was also in positive sense. And then ultimately, that one could be read directly by the translation components, and then turn that into protein. I'm just going to draw a little bubble over here, right? That worked. But what happens when it's negative sense? Well, the negative implies that it's more or less the complementary structure. It's the easiest way to kind of explain it. It's the other strand. And so let me briefly kind of highlight this guy really quick. Let's look at the little complementary strand I'm having here. And so if we had a strand that was negative, well, that one wouldn't give us the right information. If we actually transcribed and translated that guy, it wouldn't give us the correct code. So typically what needs to happen is that we need to take that complementary strand, that negative sense strand, and turn it into a positive one. So that's the extra step, in case you're wondering. Trying to get my colors over here coded. Give me 1 second. Turn it into a positive strand, and then you can fulfill the process. But even more so, you can do that with RNA as well. You can actually create an RNA version that is negative sense. And that one also, if it were to be translated, wouldn't give you the right protein. So what would have to happen is that we'd have to convert this guy into its complementary sequence, into its positive sense. Get the other color that I had similar to that. Into its positive version of it, so that that one eventually can give you the protein. And so this is where that whole decodability portion comes into play, is that some of these have this extra step. Now, what's very unique about this is that there's an advantage, is that by creating these negative templates, these negative sense versions of them, is that you can create multiple copies of the positive sense. And this is all about being efficient. You see, when you have one strand of DNA, one positive strand, or one positive strand of RNA, well, that's that one RNA that creates your protein. Fine. But if you have the negative sense and you go backwards and you create the positive version, you don't have to just create one. You can create 20 copies of the positive version. And now you have 20 templates. And each one of those 20 can create proteins too. So it's a way of amplifying things to making things more efficient, to increasing the amount of copies you can make in a shorter amount of time. And that's what also makes viruses a little bit more on the dangerous side, is that for us, every single one of our cells will require two-stranded DNA or single-stranded positive RNA, and that one copy making one more protein. Viruses, on the other hand, can go in several different directions. And so they can actually take that negative sense and amplify it, make more copies of the positive sense to make more proteins. And also at the same time, if you haven't figured that one out too, is they can make more copies of the negative sense one too, just by going back and forth. So it's a very unique concept that we'll start looking into as we start describing how these viruses work. So we'll start taking a look at them in a couple of the next steps. So now, putting that kind of bit of information off to the side, that was just one level of how to classify our viruses. Well, what about the hosts? How do we sort them out based on what they infect, right? And so what's interesting is, well, how does it find a host? How does it know where to get in? And this is a topic that is kind of important for us right now in terms of how do you select a specific host? And so once we get into part 2 of this, we'll start discussing how does each virus find its host, identify it, confirm that it's where it wants to go in, and ultimately go in. So we'll leave this for the second portion of our recording.